Welcome, everyone. Thank you so much for joining us today for our very first Fresh Fair webinar. Today's webinar is called Sparking a Conversation about Fair Data Sharing. My name is Kira Bradford, and I am a co-PI of the HEAL Data Stewardship Group and moderator for today's panel discussion. Next slide, please. So here's our agenda for today. Oops, here we go. This webinar will consist of a panel discussion followed by live Q&A. If at any point you have a question, please feel free to use the Q&A box below, and we will do our best to address as many questions as possible towards the end of the hour. Please note that this webinar is also recorded, and the recording will be made available to all registrants who registered. Next slide, please. I am joined today by 4 investigators from the Stimulating Peripheral Activity to Relieve Conditions, or SPARC community, and each of them are data superstars in their own right. It is my pleasure to introduce Charles C. Horn of the Hillman Cancer Center of the University of Pittsburgh. Dr. Horn's primary research focus is the neurobiology of vagus nerve signaling in health and disease, and more generally, the role of gut-brain communication in homeostasis. Numerous medical treatments and diseases affect gut-brain interactions to elicit nausea and emesis, reduce food intake, induce inflammatory responses, and modulate pain. His laboratory conducts studies on the impact of cytotoxic chemotherapy agents, surgical drugs, and chronic conditions on these responses. This line of research has the long-term goal of developing treatments to decrease symptom burden and improve quality of life for patients. We are also joined by Bhavesh Patel of the California Medical Innovations Institute. Dr. Patel completed his undergraduate studies in industrial and mechanical engineering in his home country of France before joining UC Berkeley in 2011 through the MS-PhD program in mechanical engineering. He is currently the PI on an award from the NIH, where he is leading the development of a software called Software for Organizing Data Automatically, or SODA. This cross-platform desktop software is intended to simplify data organization, curation, and submission for researchers receiving funding from the NIH SPARC initiative by providing an intuitive user interface and automating repetitive and time-consuming tasks. We're also joined today by Bartek Raiva of the Bindley Bioscience Center at Purdue University. Dr. Raiva is a research associate professor of computational life sciences, where he is conducting studies on the technology of high-throughput cytometry, high-content imaging, biological image analysis, biological pattern recognition, and applications of statistical machine learning and analysis, biological pattern recognition, and applications of statistical machine learning in cell biology, cancer research, and bioengineering. Dr. Raiva's expertise is in the use of modern data science and biological research. He is particularly interested in integrating biological data from a variety of linked single-cell technologies, from flow cytometry through biological imaging to proteomics and functional genomics. And finally, also on the call with us today is Susan Tappan, a scientific director at MBF Bioscience. Dr. Tappan is a developmental neuroscientist with a keen interest in stereology and other methods of quantification. At MBF Bioscience, she serves as the scientific director, leading research efforts through NIH, DoD, and other funding sources. Her passion is identifying practical solutions to biomedical analysis needs that balance the essential requirement of reproducibility as well as person-hour effort. Next slide, please. So now that I've introduced the panelists, thank you panelists for joining us today. I will hand it over to Dr. Jessica Masrick, the director of the data ecosystem for HEAL, for some introductory remarks. Jessica?
Hi, everyone. Thanks so much, Kira. Thank you to the stewards for organizing this, and to the SPARC PIs for joining us to share their knowledge today. HEAL is a trans-NIH effort to rapidly produce research-driven solutions to the opioid crisis, including addressing pain and pain management. HEAL has a highly diverse research portfolio that ranges from basic science to medication development to implementation science. And we really do cover 6 major areas that are part of the HEAL initiative. These are translation of research to practice to treat opioid addiction, enhanced outcomes for infants and children exposed to opioids, preclinical and translational research in pain management, clinical research in pain management, novel medication for opioid use disorder, and new strategies to prevent and treat OUD. As we've been working hard in the HEAL initiative, on these research portfolios, we've heard from our stakeholders that the data and results from HEAL studies in all of these areas need to get out to the community to augment our research findings and accelerate the solutions for OUD and pain. Can you go to the next slide, please? Perfect, thanks. So in response to this need for broad sharing, we're building the HEAL data ecosystem to disseminate HEAL results and data. And we're taking a user-centric approach that attempts to account for the variation in HEAL studies and data, and also address the diverse needs of potential users of the data. For example, we know that data can be valuable to future research studies and will be accessed by researchers within and outside the HEAL initiative. But we also understand the need for the broader community to have access to data and results. For example, clinicians, advocates, patients, people in recovery, other government agencies, and other stakeholders. So the infrastructure we're designing to provide this access is the HEAL platform, a web-based interface to search for studies and data and access data and information, and even do analyses and access tools and workspaces. The platform, which is being built by the University of Chicago, will also provide access to studies, publications, and data being generated by HEAL investigators that are working on these really diverse research challenges. We're building this with the goal of making it accessible to a broad audience, again, because we've heard of the importance of it from our broad group of stakeholders. And to make this infrastructure possible, HEAL data need to be made fair, findable, accessible, interoperable, and reusable. And the stewards are providing expert engagement and guidance toward this goal. They're developing resources and compiling tools that will help HEAL researchers make their data fair. For example, they're hosting webinars like this one for people to come and learn and hear about our goals and efforts. And while we're working on this for the HEAL initiative and to meet our specific needs for HEAL, we're also aligning with broader NIH efforts being led by the Office of Data Science Strategy to put infrastructure and resources in place to make data fair across NIH. All of these efforts are in line with and will hopefully help support and prepare everyone for the new NIH data management and sharing policy, which will go into effect in January 2023. Now back to today's big event. We're excited to have you here. We're excited to dive into FAIR and the importance of FAIR in this webinar now. And I know that it will spark interest, engagement, and continued conversations. I'm looking forward to being part of all of those. And now, Kira, pass it back to you.
Thanks, Jess. All right, so we're going to exit out of our presentation, and I invite my panelists to please show your camera so all the audience members can see you as you answer your questions. Thank you. All right, so let's do some level setting. Bhavesh, let's turn it over to you. Can you help us understand what is FAIR and what are FAIR principles?
Yeah, sure. Hi, everyone. I'm Bhavesh. Thanks for having me. And so the FAIR data principle, where FAIR stands for findable, accessible, interoperable, and reusable, were introduced in 2016 by a group of diverse stakeholders representing academia, industry, funding agency, and publishers. They include 15 principles that everyone should follow when curating and sharing their data and metadata, such that they can easily be reused by humans and machines, and do that without help from the creator of the data. And so typically when we need data from a study, we have to email the authors, and more often than not, we never get a reply back. And if ever we manage to get our hand on the data, it's typically very difficult to understand and let alone reuse the data or reproduce the results. So the FAIR data principles are intended to address such situation and many others that inconvenience potential reusers of your data. And with the advent of machine learning and artificial intelligence, the FAIR principle also encourages to curate and share data sets consistently, such that they can be automatically injected into ML and AI pipelines. And so the principles are purposely designed to be very high level, such that they apply to any data type, irrespective of the nature of the study or your field of research. For example, a couple of those principles state that data must have rich metadata, or data must be shared with a clear and accessible license. And it is then left on the researcher, the lab, their funding agencies, or the community to decide how to precisely implement each of those principles such that they are consistent or applicable for their data type or the type of study that applies to them. For example, in the SPARC program, all data must be shared, must include some Excel file that includes metadata such as information about the contributor, the species used in the study, or protocol of the study, etc. And all of those data sets must be shared under the Creative Commons Attribution License, or CC-BY, which allows reuse by anyone without restriction, as long as they credit the original authors of the data. And I would recommend everyone to read the original FAIR paper for more details, which is an excellent starting point, I think, to learn about the principle.
Thanks, Bhavesh. We'll be sure to include that as a resource. So let's think more broadly. Why should the scientific community care, and how would they benefit from applying FAIR principles? Bartek, let's hear from you.
Well, there is a short and a long answer. So the short answer is that FAIR is just the manifestation of everything that is great about science. And the longer answer would be that if we look at scientific principles sort of formulated through philosophy, what we call Mortonian norms of science, right? We have organized skepticism, which is basically the fact that we engage in peer review and we test our ideas through rigorous processes. We have lack of conflict of interest or disinterestedness, right? We have universalism, and the last attribute of science that Merton defined was communalism, not to be confused with communism, of course. So communalism is the common ownership of scientific ideas, right? Now, if we keep this in mind, that common ownership of scientific ideas and discoveries is one of the key, I would say, pillars of science, then we have to ask ourselves, okay, so how scientific data is related to those concepts? Well, let's look into the definition, and actually, if one pulls the definition of what is scientific data from the NIH document defining the data sharing policy, right? They came up with a definition, so I looked it up, and the definition in first sentence says that scientific data is the recorded factual material commonly accepted in the scientific community as a sufficient quality to validate and replicate research findings, right? And so on and so on. So I just read it from the actual document. Okay, so if we know that scientific data is the material required to validate and replicate research findings, and we know that the fundamental pillar of science is common ownership of scientific discoveries, and is organized skepticism, which is rigorous testing of the ideas, none of those two can be executed. We cannot engage in them if we don't have ability to validate and replicate research findings. So in a sense, I would say that we always had a concept of FAIR somewhere in the background of organized science. The only difference is that recently, with the massive amount of data that we produce, this concept was, I would say, codified, defined, maybe better, and we started to develop tools allowing us to share the data. But it's not like we are talking about anything new. We are really talking about the fundamental background in which, or the fundamental structure in which science operates. So the answer, to cut the story short, the answer why the community should care about FAIR, well, because the community of scientists should care about science, and FAIR is just the key ingredient of a scientific process.
Thank you, Bartek. All right, so that was a broad perspective, but let's narrow it down specifically to the individual investigator. How would I, as an investigator, directly benefit by making my data FAIR? Let's have Charles and Sue take a shot at this one.
Thank you, Kara. So one thing that you might be struck by immediately is this is an additional burden. It's on my shoulders. Now I have to start putting my data, my procedures, my code in the public domain and have to organize them. But I would argue that it's a secret weapon because you can make your processes more available. You can make your research more credible, more visible, your work, in at least two ways, one of which is for grant applications. So just to make this concrete, you can make digital object identifiers in a reference list and document your data, your procedures, your code throughout the body of your application. And we've done this in the SPARC consortium using the SPARC portal and generating DOIs for all of our data sets that go into there. And we can reference those data sets in the future. And also you can show a well-defined process for data and resource sharing in those sections of your application. So that's for grants. And then secondly, for publications, you're showing the accessibility of the data. The code are often requested as part of the submission or the acceptance process for the journals that you submit to. It's becoming very common now. So in having that up front, I mean, you don't have to do this post hoc dance after the fact and trying to find a place to put your data, your procedures. You already have that in place. And I would also argue that manuscript reviewers are looking for that in their review process. So this only boosts the manuscript credibility. And in general, I think staying ahead of the curve, and I would argue staying competitive in these aspects is a very good idea because a lot of labs are following this or are moving towards this. I'll turn it over to Sue. I know she has something to add.
Thanks, Charles. I think what you've said really speaks to the consolidation of the data process of taking the experiment from inception all the way to publication. So sharing our interpretation of data, but the DOI process of adding identifiers and citation ability to not just the manuscript, but also the data source, as well as the mechanisms that you use to create the data, allow you to generate a better conversation and a better awareness of the effort that you're putting into every stage of the experiment, not just the interpretation of the data, but also the generation of it and the analysis of it. So I think that those things allow, especially early career stage investigators, so your graduate students, your postdocs, your early stage scientists, an opportunity to demonstrate a track record of how they're building their career of not only in manuscript form, but also in the operation of their critical thinking. So the skills that they're building for the data source. The other thing that I really think adoption of FAIR principles brings to individual researchers in their lab groups is greater efficiency within your local group. So it can make the process of bringing people into your lab, getting them up to speed and productive to your procedures, your research plans and strategies early by having well-documented protocols, a formalized mechanism for how you're going to name files and how you're going to store them, all of those sorts of situations that you have a basic process for, conceptualizing them from not just an individual research project, but to a principle for your entire lab or lab collaborator group, just makes it easier to share information and make people more independent early. And so it makes that process of going from a lab book of here's what I did for this particular experiment now, just take over to demystifying it. So taking, another analogy might be you get a representation, a recipe from your grandmother. It's handed down through generations. You know that your grandmother made the best biscuits in the entire world, and no one can convince you otherwise. But try as you might, you can't quite get that magic secret sauce. And it's because there's more to the biscuit than the ingredients. It's the procedure, it's the process. And so you learn that in grandma's kitchen by watching her actually, create the biscuits. You learn that mechanism. And so the same goes for protocols. You can do that in your lab. And by formalizing that procedure early and often and revisiting it and updating it as you go allows you to, bring people in and allow people to leave with confidence, so that when your graduate student moves on to their first postdoc, you don't have that existential dread of wondering whether or not the next person can go. So go on and expand in their research program. So that's what I would say is a mechanism for how this helps your lab.
Thanks, Sue. I think we have heard a very firm argument for making our recipes fair in addition to our data fair. And I have definitely experienced that existential dread when a previous grad student has left the lab. But let's move on to our next question. So what are some examples of groups or teams seeing a benefit as a direct result of taking on the effort of making their data fair? So we'll start off with Bartek, and then maybe Charles and Bhavesh have some examples to share.
Yes, so Bhavesh mentioned this at the very beginning, that SPARC program, NIH SPARC program had FAIR principles kind of built in from the very beginning. And it's a very big program with many, many researchers. So my personal experience was that, and I was, when I started participating in this program, I was actually a newcomer in the field of neuroscience. My participation was mostly due because I provided kind of complementary knowledge on the methodology that was used by neuroscientists. So I didn't know a lot of people in the community, as you can imagine. But the fact that there was this expectation and the mandate to share the data, that allowed me to access others people's data fairly quickly. Not only learn about what they are doing, but test my methods, which I'm developing machine learning methods, statistical methods with this data. And eventually this led to a number of new collaborations, which I don't believe would ever happen otherwise, because not being familiar with the community, not knowing people, I would have problem probably accessing this information otherwise. I wouldn't know what I would be looking for, right? I wouldn't even know what type of questions I should ask. But because the publications were accompanied by the raw data, or the raw data is very often released within the community even before the publication happened. This allows us all to form those ad hoc new collaborative groups, and this leads to really better science, and we can rely on the complementary abilities of the people in SPARC community.
Just, I guess it's my turn, but so to take off on that, I would say that there's a temporal aspect to it as well. Just speaking about SPARC, we've had several mechanisms that have been rolled out over time in SPARC the last 5 or so years, in which the early awards were given the mantle to collect data, and they collected a lot of data. For example, there's Terry Powley's group at Purdue, which Bartek was a part of, and they generated a lot of data on the gastric, the GI system. And then later, we had awards for modeling and simulation efforts that use some of that data. And I'm actually on one of those teams. So there's sort of this generational jumpstart in the processing of data and the usage of data, at least within SPARC. That's my example.
That is really awesome, Charles. What about Bhavesh? Do you have any stories to share?
Yeah, one personal example I can share, I mentioned, is my team's project during the recent SPARC hackathon, where we developed an automated knowledge discovery tool for SPARC datasets. So my team won the hackathon, but I don't think this is relevant to the discussion here. But we were able to develop a prototype during the 2 weeks of the hackathon, where we show that it's possible to automatically find relations and compare multiple SPARC datasets, even if they are developed independently by different SPARC groups, simply by leveraging their FAIR characteristics. So all SPARC data sets are findable on a specific location. They all have the same metadata, follow a strict folder and file structure. So it's very convenient to mix and match all of that and run ML and AI pipeline to make new discoveries that would otherwise be, if not impossible, very tedious and time consuming. And so this shows how the pace of discoveries can be increased within the SPARC program because of their FAIR practice. And this can also set an example for everyone else to follow.
Yeah, it sounds like an incredible experience. So as part of the HEAL Data Stewardship Group, we have been engaging with many of the study investigators and trying to identify some challenges and some potential opportunities. But there is one challenge that we would love to hear feedback from this SPARC group. So we've had investigators tell us, I have very limited funding, I have very limited time that I can allocate towards making my data fair. What can I realistically do? Sue, let's start off with you.
Yeah, this is something that I think a lot about, as a device maker and a software solution creator. We need to make it easy for people to get started with data collection. And so I spend a lot of time thinking about how to be efficient with people's time. And the thing you can do right now is just focus on the devices and the current workflows that you have in your lab right now and that you use every day already, and see if you can go beyond the default setting that is set up in your microscope or your image acquisition software or even your other device equipment, and configure them for the experiment that you're doing. Input what channel is being used to image what protein label, for example, if you're doing immunohistochemistry. That will write automatically into the metadata for you, and you're utilizing your current equipment. You're making it work for you, and that allows you to know that you're not having to go back and redo things, first of all. And then second of all, it's going to be done in a very consistent and uniform way. Choosing that strategy for your naming convention within your lab so that individual lab members use the same strategy and the same folder structure even for how they categorize subjects and data within acquired from that subject. In our case, for the SPARC program, we follow the SPARC data set structure, which, as Bhavesh mentioned, has a rigorous, folder structure. But within that folder structure, it's very flexible to allow you as an individual researcher to put the data in a strategy that works for you. So there's some very simple things that you can do just to codify what you're already doing already and make your equipment work for you. Use their existing strategies to capture that metadata that you are writing down in your lab notebook.
That is an excellent point. I, as a researcher myself, I don't think I ever moved beyond the default setting. So definitely a good one to consider. Bhavesh, did you have any other thoughts for us to consider?
Yeah, I think if funding and time are a limitation, the easiest approach is to simply share your data with as much metadata as possible in any of the open repositories that are available and help you make your data fair, such as Zenodo, Figshare, or Dryer, to name just a few. If you Google any of them and add fair data or fair practices, you will land on their page where they explain how you can upload their data on their platform and provide necessary metadata and information to check all the 15 fair principles in doing so. And then if more stringent guidelines are imposed, for instance, by your funding source, you must request that they provide you the necessary support to implement those. For instance, in the SPARC program, we have the DRC, the Data Resource Center, and Sue's team is part of that, and they provide us necessary help, support, and guidance to implement those fair guidelines. my team is also developing a tool that guides researchers step by step into making their data compliant with the SPARC standards. And so the burden of making data fair should not fall only on the researcher if more strict guidelines are expected to be implemented successfully, I would say.
I really like that, Bhavesh. I feel like this is actually a good opportunity to share that for NIH and program officers. It might be time to consider what kind of support we can offer to all the studies for helping them make their data fair. Because I think I agree with Bhavesh. I don't think it's squarely on the investigator to make the full effort in that. So the next question is another question that we received from the investigator community. So this question is, I'm worried about sharing intellectual property and proprietary information. Given this, what data is left for me to contribute? Let's have Sue lead us off.
Well, Kira, I think you led right into it very nicely. And this is where NIH and your organizing groups can have procedures and policies in place to help support the research teams that they're funding to generate this fair data. Having clear citation policies, education of strategies, and assistance to help researchers develop these enhanced tool sets and strategies is really helpful, as well as ensuring that embargo periods for sharing sensitive or proprietary information, early information, so that you can publish your findings from your data as you expect to, are critical for that. Scientists can also help by citing the data sets that they use, as well as the protocols that were used to generate the data. So not only do you cite your own manuscripts and the manuscripts that you cite in your papers, but you can cite the data that you used and protocols that you jumped off from in order to create your data. These are citable objects. Drop them in your manuscript, drop them in your grant proposal, like Charles was saying, and help increase the awareness and visibility of all of these projects that are relevant and intellectually encouraging.
Bhavesh, did you want to add anything?
Yeah, I'd like to add that being fair doesn't necessarily mean sharing everything openly without any restrictions. The FAIR, as I mentioned in the beginning, the FAIR principle only state that all data must be shared with a clearly defined usage license. And then it is up to you to define and choose that license or define the restriction on the usage of your data. And if actually FAIR is helping you towards properly sharing proprietary data with a proper usage license, so it's actually helping you sharing those data where you want to put restriction in a manner which help you protect that information and define your rules for the usage of the data. So even if you have proprietary data, you should actually be looking into following those FAIR principles, I would say.
That is actually a really important distinction. Making your data fair does not mean making it freely open and available, but it empowers you to make decisions about exactly how accessible your data is. Bartek, you look like you have something to say.
Yeah, I think that in general, those fears that some people express regarding proprietary data, they stem from sort of misunderstanding of what is really what kind of data should be kept under wraps and why, at least in the academic environment, right? So the only scenario one could imagine that would play a role would be if there was some invention and the university or some other academic institution, research institution would decide that they want to patent this invention. Well, of course, then you have to talk to your technology transfer officer at your institution, but you will learn two things very quickly from them. Number one, data itself just raw data is not patentable, right? Abstract ideas are not patentable. You have to have an actual invention. And then when the invention is disclosed, when the university actually files for a patent protection, then again, I love to read from the source documents. If you go to US Patent Act, you will learn that any description of invention, so if you file a patent, right, shall be provided in a clear and detailed manner to ensure that any person with ordinary knowledge and skill in the technology sector with the relevant information can recreate the invention. That's the whole deal, right? That you get a protection for your invention, but it has to be described in such a way that it can be reproduced. And now we are coming back to the definition of scientific data. What's scientific data? Well, according to NIH definition, scientific data is the information of sufficient quality to validate and replicate research finding. So if you submit a patent, then after patent submission, when the provisional number is assigned, you not only should, but there is absolutely no obstacles, right? You should actually share your data through the FAIR mechanism. And in any other situations, you should, because it's good for science and it's good for you. So I don't see a conflict here. And I think that people who are worried about how this plays out in the context of possible inventions and patents, they should definitely talk to their Office of Technology Transfer officers in their institutions, and they will find out that it's much simpler than they thought.
I really appreciate that perspective, Bartek. So we're going to go on to our last question. So let's say we've sold some investigators today on FAIR principle. So taking that perspective, they feel particularly motivated on starting on making their data FAIR today. Where would you recommend they start?
Okay.
And let's have Charles. Yes.
So you're convinced we've sold you. So what I'd like to say is there are some low-hanging fruit that you can implement. And what I mean by this is low cost, low financial cost, and low effort on your part. And in fact, you can think of it this way. You can roll it out to your team and have your trainees do it, okay? So the things that I would recommend would include GitHub. I think GitHub's a really good resource for hosting code, for hosting documents, for hosting quite a few things in which you want to see revisions, you want to see the changes, and they have free accounts. It's owned by Microsoft. Very steep educational discounts if you want to get all the bells and whistles. So I think it's a really excellent platform for putting those things up, especially text-based information. And then the second thing, I'm just going to reiterate what Bhavesh had said about some of the databases where you can stick your data, one of which is Zenodo, which is hosted by CERN. It's a European physics consortium. It'll provide you DOIs for submitted data, protocols, whatever you want to put in there. And you can also link it up to GitHub. You can actually take a flash shot of your GitHub repository and you can store it there and then have a DOI.
So hang on, Charles, wait, what is a DOI?
Digital Object Identifier.
And how does that make my data FAIR?
Well, you can cite it. Okay.
Ah, okay.
And you get a link. So you can put in a URL somewhere in your reference list, and you can click on that and go to that data set or that repository or whatever has a DOI. So that's a really great thing to have. Lastly, I would say something, and you'll have to check into this with your own institution, but here at the University of Pittsburgh, our library has taken really a lot of steps forward in making things available, trying to train people in FAIR principles. For example, we have our own data repository. I keep telling those folks that I'm putting my data in SPARC, in the SPARC portal, and I'm not using them right now, but they do have that. They've already set that up. They'll also sit down with me and orchestrate a data management plan. So the librarians have actually taken this task on. They have software training in GitHub and Python. A lot of short sessions that you could take in an afternoon, or you can send your students over to take. Obviously, we're pressed for time as principal investigators, and you don't have time to take a whole course in Python or in data management, but you can get this from the library. So you should check with your library to see if they have these resources available.
And Bhavesh, did you have any other considerations?
I think Charles mentioned pretty much everything. I think if you're sold and want to start, again, as I mentioned, I would say read the original FAIR paper, start using those repositories. And similarly to Charles, I also develop code. So what I do is work from GitHub. You can link your repository with Zenodo. So anytime you make a release of your code on GitHub, it automatically gets archived on Zenodo. And the DOI, besides what Charles mentioned, also give you a unique identifier for your release. So it makes it also findable, easily distinguishable from other resources and uniquely citable, as Charles mentioned. And again, personally, my team is developing tools for SPARC, but we're starting to develop tools outside too to help researchers make their data fair. So I'll be happy to discuss with any of you if you reach out as well. And as would, I think, any of the panelists, as you saw, probably we are all passionate about fair data here.
Well, we're going to put that passion to the test. We're now going to start the live Q&A session. So we'll take a look at the first question. All right, so data, tools, biomedical domain in the general sense, these are concepts that translate into certain regulatory requirements. Have you looked into pathways for regulatory approval of the data-driven approaches and tools as devices in a general sense for regulatory approval by the FDA? Let's have Sue start off with this one.
Well, in short, no, I haven't looked into taking any of our tool sets through the process of regulatory approval for the FDA. So I can't explicitly speak to the strategies where these data-driven device strategies, approaches are directly link into the FDA regulatory pipeline. However, I do know that in order to submit a device for FDA regulatory approval, you must have GLP, you must have everything documented and have change control procedures in place so that every modification to your device, its output, is documented and ready for inspection. I don't know if Bhavesh or Charles or Bartek have any direct experience with the FDA process.
Well, I do not, but I think that this is a very interesting question for another reason, because it points to something that we haven't discussed, and that is the emergence of the processes or devices which are data-driven or based on, let's say, machine learning, like computer-aided diagnostics and stuff like that. We haven't really discussed this, but indeed, I hope, and I think that we should all hope, that at certain point it will become a requirement that the training sets for all of those devices which end up with the FDA approval will be fair shared. Right? So we know how those devices operate, what motivated them to operate in a particular way. This also relates to the very hot topic in the biomedical community of explainable AI, right? So I didn't look into this. After this question, I'm actually much more interested into looking into this, but I think that the step from sharing data through FAIR processes in scientific community to requiring that those FAIR processes are embraced by the by FDA should be made at certain points, certainly.
All right, I think we're going to move on to the next question that we have. Any advice to heal investigators on achieving fairness, as many of our projects are from individual investigators in smaller research labs, not necessarily a part of large consortium or programs such as SPARC? Let's have Bhavesh start us off.
Yeah, definitely. I think I'll go back to what Charles and I mentioned earlier. You can start by simple step of using GitHub if you're developing code, or if you have data. Look at the pages of Zenodo or Figshare where they explain how you can upload, add metadata, and share your data on their platform. And at the same time, again, check all of those 15 guidelines of the FAIR principles. So that would be my suggestion if, again, time and funding are a limitation or resources are a limitation.
If I could jump on as well, for researchers who aren't necessarily writing code and writing their own analysis algorithms and processes, you can utilize existing tools such as SODA, which Bhavesh is creating to help create a manifest that describes what your data is. Just because it's not in use or it was made specifically for SPARC doesn't mean that you can gain some usefulness out of that tool. Likewise, we have created through the process of SPARC a software tool to add metadata to your microscopy image data as well. So if you haven't configured your microscopy device to write all of the rich metadata that those devices actually have the ability to write, you can add it after the fact. We've created a tool to help streamline that for you. So I think there are tools that are coming into creation, not just through the SPARC program, but through other avenues as well that help actual data generators create not just the source data, but the data that describes the data, the metadata. And that can go right all the way through to your protocol. So in the SPARC program, we use protocols.io, which allows you to document the procedure of what you do in your wet lab to actually create the data. You can get a DOI for that. You can make that public, and you can branch off of that protocol for each change that you make. So let's say that you change the dosing regimen for the fill state of your device. You can just spawn a new protocol without having to rewrite all the steps. You just spawn it, you clone it, you create the change. Now you've got a DOI, carry on with your day. So there are simple things that you can do right now. It's just the activation energy of making that first step that seems hard, I think.
Thanks, Sue. We'll move on to our next question. Are universities and research centers using F&A funds to support and expand use of electronic notebooks to facilitate adoption and use of data and metadata standards? So who thinks they have a perspective on this? Bartek?
Yeah, so I think that the first issue which has to be mentioned is that laboratory notebooks, preliminary analysis, case reports, drafts, all of this is not what FAIR is about. In fact, again, I'm going back to the NIH definition of scientific data, which explicitly says that scientific data does not include all of those things. So your laboratory notebook is not something that you have to share. On the other hand, one may argue that if there is some mechanism for electronic notebooks, then it would be easier to later on take a part of this notebook and convert it into a shareable object. Well, that's sort of a separate issue. But in general, from what I see, universities, whether using NIH or NSF or other funding or their internal funding, they are building their own data repositories, which allows researchers who have data that are not yet published or they do not really belong to any of the existing repositories, put them in one place and get the DOI number. Whether those repositories are going to be sort of compatible in a sense of data transfer with electronic notebooks run at given universities, that's a matter of a choice for the IT departments in those universities. I didn't see any policies, and please correct me if I'm wrong, mandated by NIH, which would mandate anything like that, because as I said, NIH specifically said that laboratory notebooks are not the data that should be shared or that needs to be shared, right? You may share your notebook if you want to, but it's not required.
All right, so we have another question. I read the new NIH data sharing policy. I believe that for NIH funded studies, it was guiding investigators to share data on discipline-specific NIH repositories. Is this your understanding? I would actually like to invite Dr. Masrick to be our 5th surprise panelist to answer this question.
Hi, thanks, Kira. Sure. So yes, if that repository exists for your data, then that would be a great place to put the data. But in a lot of cases, that repository might not exist. So if you look further down in the policy, it has a couple of other suggestions, such as submitting data supplemental material to PubMed or choosing a generalist repository. There's also a supplemental notice that goes along with the policy for data management and sharing that talks about, where is it? It talks about general principles for choosing a repository. And so that might be helpful to look at as well. And then for HEAL investigators, I don't want to give any spoiler alerts here, but we will have resources on that the stewards are developing on the website that they host, HEAL Data Fair, to help guide HEAL investigators' decisions around repositories. And that'll be coming soon. I don't know if Kira, you want to say anything else about that?
I will in the conclusion slide, but I also want to ask Bhavesh to give a more investigator perspective on the NIH data sharing policy.
Yeah, I think, I'm not sure if this was mentioned, but the new policy that will, I think, go in place January 2023 will require with all applications to the NIH to have a data sharing plan that explains how you will make your data fair, and that would be irrespective of the amount of the grant. So that's another reason for you to get onto that fair train. And the other thing about using data-specific or general repositories, I think we've been saying a lot about those general repositories, and of course, in no way you should share on those repositories if there exists a specific repository that applies to your data set, or if it is required by your funding source to share on a specific data repository. This is the preferred way, of course, always. Into FAIR, you have findable, and one aspect of findable is to share your data where potential reusers are expecting them. And so the preference is always data-specific repositories. And again, in the absence of any requirement or support or specific repository, then only you should go look for those general repositories that makes it a little easier for you to share because they are not as restrictive as those general repositories, data-specific repositories.
All right, so this looks like it's the last question from the audience. Sue, I think this one's for you. What is the name of the tool for adding microscopy metadata?
Yeah, so the tool is called Microfile Plus. I've added a link into the chat, and I think we're sending out a list of resources based on our conversation today. So I'll include that. It's free to download, it's free to use, and it accepts proprietary microscopy image sources. So Zeiss, Leica, Nikon, any of the major brands, as well as TIFF files that you may have created from your own homegrown microscope. And it allows you to add any relevant metadata that you want to make sure your data file, your image file contains for posterity going forward, not just within for sharing, but within your own lab. Makes it easy.
Awesome, thanks, Sue. And yes, let's bring up the last slide and we'll do closing remarks. There we go. All right, so first, thank you everyone. We really appreciate the time of our panelists and our audience today. For the audience, we hope that you are leaving with a better understanding of FAIR, why the HEAL initiative is emphasizing FAIR, and how it might benefit you as investigators. So in addition to the recording of this webinar, we will also email who has registered to this event a list of resources that was mentioned during our call today to help assist you as you participate more deeply in the HEAL ecosystem. We also hope that you will reach out to us, the HEAL Data Stewards, for questions or support as you embark on your FAIR journey to maximize the value of the HEAL Data Initiative. And a link to our Slack channel and our Connect with Us forum are going to be located in the chat momentarily, and we will also share those in the resources to be emailed to you. Thank you all again so much for participating for our very first webinar. We hope you all will save the date for our next webinar on October 21st from 1 to 2 p.m. Eastern Time. Just a little sneak preview, we'll be discussing how to make thoughtful data repository choices. So if agonizing over which repository you want to put your data on is keeping you up at night, stay tuned for this next webinar. And with that, I adjourn this meeting. Thank you all so much.
Thank you.
Thank you.